So you have, these are the binding pockets of the ribosome. So, you know, there is, the ribosome is like positioning everything to do this hydrogen bonding and stuff. The driving force. Okay, so the ribosome looks like this. There are, so E. coli alone, you know, which is a little less complicated maybe than the eukaryotic cell, there are about 20,000 ribosomes at a given time in a cell. A growing mammalian cell, I love how it says growing, so like dividing or something like that. They have around 10 million on average in any given time. So 10 million ribosomes. So we have, in the first image, we have a picture of the 70S ribosome, which is in the prokaryote, and we have the 80S ribosome, which is in the eukaryote. The S refers to sedimentation. It doesn't refer to like how many amino acids are there. It doesn't refer to that. Or it doesn't even give you like mass. It's not really like a, you know, molecular mass like you would think. It's sedimentation value because it's like, remember cell fractionation, we can fractionate the cell, spin in a sucrose gradient, and where does it sediment at is where these numbers come from. So the prokaryote has a smaller, it does have a presumably smaller ribosome. It has in total 70S. The two pieces that come together, the big unit is the 50 and the small is the 30. In this diagram now we can really start to think about, we got this picture of RNAs and proteins and that. We have 23S and 5S ribosomal RNAs and then 34 proteins that build this structure right here. And the 30S is 16S RNA, ribosomal RNA, and then 21 proteins in there. The sequence is different from what the eukaryote might have. There might be some conserved things in there, and then there's additional things in the eukaryote, how it works. But the sequence is not identical. There is something called homology, which means that the idea of homology is that sequences come from the same ancestor. So if you have a homologous sequence, that means they came from the same ancestor. It does not mean they're identical. Okay, so the eukaryote is 80, and we have a 60 and a 40 for it, and there's 28S and 5.8S and 5S ribosomal RNAs. So there's when you see these differences, this means that there's like different chains. There are these different chains of RNA in there. So there's more than one chain of RNA. And then we've got 46 proteins to make that complex. The small subunit is 18S ribosomal RNA and 33 proteins. So an actual like X-ray crystallography picture of what it looks like right there in the blue and the green. And I got it. My clicker's going crazy. I don't know if that's all, I mean, so you can see, it's really dark, but you can see the RNA is kind of the ribbon stuff, but you can see like these little balled up things, and those are the amino acids, the proteins in there. So it's like all of it together. Very detailed. Okay, so when we're talking about transcription, we can't get right into the ribosome and like moving in the active site. First, the picture we want to look at is, you know, how is the transcript read? And there are a couple of different ways that transcripts can be read. So there are, in prokaryotes, it's very typical to have what's called a polycistronic transcript, mRNA transcript. And what that means is there are going to be multiple protein coding sequences within a single transcript. Okay, so like on this one, we've got that green protein coming off of coding sequence for protein number 1, and then we've got protein 2 and 3. Notice that there are these little red arrows are showing us that there are multiple start sites for the protein. So where the where the ribosome begins, there's 3 of them on this single transcript. The eukaryotes are typically monocistronic, which probably is good because we have all that intron processing and all of that, right? Which means that a single transcript belongs to a single protein. So the other things that while we're looking at the RNA, here's our eukaryote. We got our 7-methyl guanosine cap on the 5' end and the poly A tail on the 3' end. We also don't forget that we have these untranslated regions. So UTR, that's what those are, untranslated regions. What does that mean? That means that they don't carry 3-letter codes that are read by any tRNA. Okay? But they're not garbage, they do have their job too. Okay, so now looking a little bit closer at our ribosome and how it enters and all that stuff. Our prokaryotes, our prokaryotic mRNA is here's the mRNA that's folded in that the complex is in blue. No, sorry, I'm doing this wrong. The ribosomal RNA that's folded into the complex is in the peach color, and then the actual mRNA that we're reading is in that blue color. So we have the SD sequence or the Shindell-Garneau sequence in prokaryotes, and this is a spot that will hydrogen bond the highly conserved sequences in our prokaryotes. Where is this at? Where is the Shindell-Garneau sequence at? We've introduced it a long time ago, and it is in the untranslated region. So that's not garbage, right? So it's in that untranslated region. And so what is this doing? It's allowing for that initial binding of the prokaryote mRNA with the ribosome. It's allowing us to bind it. Okay, so the eukaryote does something a little bit different. Here we have our 40S ribosomal subunit, and we have a 7-methyl guanosine cap on our transcript. So the 7-methyl guanosine cap actually has a binding position with the small subunit. So notice that these things don't bind the fully formed ribosome. They don't start off that way. They bind part of the ribosome, one of the subunits. So we have that initial binding, and then this thing can, once we interact there, the small subunit scans along and finds the start codon, the AUG. Okay, so we'll end with this translation. Well, so we'll come back with the translation overview actually on